We will then move the subject slightly to perhaps what many of you find of very much interest, which is we will take you through the story of how this fish AI prototype was developed during the course. And as Fulvio briefly introduced, we have 3 different parts of this work, which contains the cells that will be covered later, and also the in vitro digestion and the device itself. So the plasticware in my head. And we will start with the device, which will be presented by Pedro Costa from Biofabrics. He is a founder and CEO, CTO of Biofabrics, which is a company specialized in the creation of 3D biotissue analogs. You are a biologist by training, an engineer or maker by the heart, you say. You did your undergraduate studies in applied biology, followed by a PhD in biomedical engineering, and has specialized in the combination of highly automated tools, such as 3D bioprinting and bioreactor technologies in the field of tissue engineering and regenerative medicine. So please, Pedro, the floor is yours.
Okay, so it's working? Yeah. Okay, perfect. Thank you very much, Trond. Thank you all very much for attending this workshop. Everybody here and everybody, the huge amount of people online. That's always refreshing. So I'll be showing you what Biofabrics and also University of Ghent did for this project. And unfortunately, we couldn't have anybody from Ghent University here, but I'll be showing the work of both institutions. Actually, it makes a lot of sense because we did a lot of work together, and it actually goes together very well. First, I will show you what was the initial idea in this project. This is what the project that was approved for funding. So it was a simple idea. So basically consisted of creating a membrane with these crypt-like structures, architecture. Initially, the idea was to do this by simple molding, and then having this membrane integrated into a device which would allow to perfuse fluids, add cells to a membrane, and then simulate the wall of the intestine of a fish. So this membrane component was mainly done by University of Ghent. So they are like specialists in material sciences for mostly human applications. We are also mostly focused on human applications, but has been very interesting to also explore the field of fish as well. Also, there are a lot of opportunities here in this field, both for research and commercial opportunities. So the first thing to do, as planned, was to try to mold these gels. Okay, so they started by using PTFE molds, so rigid molds for molding certain kinds of gels. Then these molds were a bit too stiff for this application. They switched to PDMS, so silicon basically, and they could actually obtain some nice 3D structures from this molding process. However, this was done mostly using PEG-based materials, which in our case was not the ideal kind of material because they were quite too rigid, because there were a few important requirements in developing this membrane. One of them was the anatomical shape fidelity, which was achieved right away, but then there were the mechanical properties that were also extremely important in this model. And with these materials, it was not possible to do that. Then we also had to test permeability, biocompatibility. So the materials that would be ideal for this kind of application were not moldable. So Ghent came up with a very good solution, actually easier, more straightforward, just directly 3D printing these gels. So they are actually specialized in this kind of technology, so that helped. They had this already specific kind of material, which is a metac-related gel, and which has many variants, but that this general material is very good for 3D printing, actually. So we tested different things in these materials. First of all, how well does it cross-link? So how well does it polymerize to be efficiently printed? And that's when it was possible to understand that a specific type of GELMA, the GELMA AIMA, was the best one because it cross-linked faster, so the process would be much more efficient and faster. Then it was also important to add some porosity to these materials, especially so that it would be more permeable, but also to improve furthermore their mechanical properties. And so different compositions, different percentages of porogen were tested, and actually it was possible to achieve better mechanical properties as well as, and I'll show you later, better ability for molecules to go through these materials. But then after that, it was time to actually print these materials, and it was actually quite easy. It was possible to obtain very regular, very well-defined structures, villi with a height of around 200 micrometers, base diameter more or less 200, and spacing 200. So very regular, very well-defined, as you can see. But then we needed to test these other parts, so permeability and biocompatibility. And for that, we needed this second part of the device to be used, the device which was our responsibility. So, so the main idea about this device was to actually develop a dual perfusion culture chamber, so having two chambers divided by intermediate membrane, which would be permeable, which would be seeded with cells, and then act as a selective barrier to compounds of the feed, which would be flown on one component, on one chamber, and then go through this membrane and then be collected on the absorbed feed flow. This would allow to mimic the gut, and then, as expected, to study and improve the nutrition of fish. That was the main idea. And actually, very early on, we developed the first prototype, which is a typical dual chamber system, dual perfusion, being able to hold the membrane in the middle of it, with actually glass windows on both sides, so it would be easy to image the membrane from both sides, screw-based clamping of the membrane. Even we tried magnet-based clamping, which was an interesting idea, and also soft clamping, so using silicon components, so it would be it wouldn't damage the membrane, which was quite delicate. However, our end users in the project found this type of device too difficult to assemble, difficult to change the medium, and also requested specifically to have an open top system. So it would be possible to do, in particular, tear measurements with these sensors that you have for tear. So the handheld probes. So we had to basically go back to the drawing board and go to the very basics of all of this and really ask ourselves and our partners, what are, first of all, these researchers used to using in this kind of study? And then we try to make it as simple, as obvious, as easy as possible for them to use something like this. Also, it was very important from early on to make these devices as adjustable as possible, because during this process, there were many variabilities, different changes in the properties of the membrane. So it would be important to have one single device which could hold easily different types of membranes. That would also make our life much easier. And also have a device which would be easily upscalable, so having multiple devices working at the same time, and upgradable. So starting with something very, very simple, and then little by little increasing the complexity of this system. And well, we started the design process with the very basic, the most important component of all of this, which is the membrane. So we added silicone rings on top and bottom of this membrane Why? Because silicon is soft, so it doesn't damage this delicate membrane. It's compressible, so it's easy to adjust to the characteristics of the membrane, and it's watertight, so the components of the feed go through the membrane and not around the membrane. And also a very small but very important component of all this is this little ring with a very small protruding feature there, which was actually very important in this. So it would prevent the twisting and damage of the membrane. Why? Because we then adapted this insert-like device in which we just twist and lock in place the whole system, creating this watertight interface, which easily can adapt to virtually any kind of membrane. And then you can have, as intended, an absorbed feed component, digested feed component. Of course, in the meanwhile, we protected this design. And then you can actually have this, just like any insert, in a well plate and have many of these devices at the same time being tested. This is an adjustable insert system, not dynamic yet, only static. Okay, we'll see about the static later on, the dynamic later on. So with this, we could then continue with the validation of the membrane. So we did some permeability studies, and actually we could see that with the porogen, there was a greater permeability, which was quite acceptable comparing to the non-porous membrane. Also, rainbow trout epithelial cells were seated in this membrane. Then with ALP and confocal, it was possible to see that indeed the material was biocompatible, although there was something that was not perfect. So actually cells grew quite well at the basis of the membrane, but they couldn't reach and fully cover the tips of these villi in the 3D architecture. But well, still very good results. We could achieve all these requirements initially, so that was already a good start. And actually, we published this in a very nice journal. This was a collaboration of Ghent University, University of Milan, and Biofabrics. Then we actually started using human intestinal model, human intestinal cells, and I'll explain later why. So we did a similar study, but this time comparing non-porous structures and comparing 2D structures, so flat membranes, to 3D membranes, okay? And we could see that actually 2D and 3D structures have the same permeability. The seeding efficiency is more or less the same, although still not as good as a normal culture plate. And in the tier analysis, we could also see something important. We could have proper readings in the flat surface, but not in the 3D surfaces. And if you go to the imaging, you can understand why. Again, cells do cover in the 3D structure, cover the base very well, but they cannot reach this top of these villi. But still, we learned a lot from this, and we published another paper, this time collaboration between Ghent and Biofabrics. The following study was to compare all 3D structures, but in this case, non-porous and porous. And in this case, we actually got very important results. We could, for the first time, see that both in 3D structures, both porous and non-porous, we could obtain proper tier readings, which was something that was still lacking. And actually, we saw that we could have this kind of reading in the porous membranes earlier, so at around 14 days. Also, we got quite high metabolic activity in the porous and also expression of some important genes. So fast epithelial formation on the porous scaffold and enterocyte differentiation on day 35. And of course, we published this, Ghent University and Biofabrics. Last but not least, we also tested something else which was important. I don't know if you noticed, but when you use human, well, the human intestine is a bit different from fish. So you do have villi, but also crypts. So in this case, there was a new design, also adding the crypt component, which worked fine. And also some new variants of the original material were also tested. And actually, we got some very important results here. So it was possible to culture these cold cultures. Also had expression of tight junction proteins, mucus secretion, barrier formation at 14 days. Also, we could tell that the here value is determined by the type of hydrogel, not the morphology of the membrane. And this 3D morphology actually supports the upregulation of enterocyte markers. Also, very importantly, and I think this will be very relevant in the future, these materials actually have the potential to be even more physiologically relevant than normal tissue culture plates and even matrigel, which are currently the gold standard in any gel-based cell culture. So I would say very important results. And this was work done by Ghent University. They submitted it. Hopefully it will be published very soon. Now, moving on, we said that we would do this dynamic, so not just static cultures, but dynamic with perfusion. So we continued developing these concepts. We developed a lot of different designs, and we at some point decided to try to go back to the initial idea and also the first concept that we came up with. So same design process, membrane, silicon rings, anti-rotation ring, this time a bit different, which allows flow of fluid over the membrane, and you'll see how. And then adding this to a kind of insert-like system, but a bit more complex. And then you just simply twist, lock, and then, in this case, inject bidirectionally the absorbed feed into one chamber, and then the digested feed into another one. So you move fluids for having circulation of fluids by injecting, moving bidirectionally the fluid in and out of the device while always keeping a minimum amount of fluid in there. We protected this design, of course, and then you would just integrate this into a well plate, 6 well in this case, and then just move on. Although in the meanwhile, and unfortunately, we got to this a bit too late in the project, we came up with an even better idea. Why not just communicate with the initial insert system-like device, but in an easier way, and inside a well? And we basically came up with this very simple solution. We just designed a new kind of lid. In this case, a lid which has these silicon spots that you can actually flow fluids through them by using these cannula systems, which are very common, especially in hospitals for collecting blood samples. They are single-use, they come sterile. You have in one end a needle that you pinch through the lid into the well, inside the insert or outside. And then the other end, you can connect to pumps, either syringe pumps for bidirectional perfusion or even peristaltic pumps for unidirectional continuous flow. So very simple system to use. And we did a very small proof of concept study to actually show, well, test if this setup would work in the daily routine in the lab. And we actually did a small study with these devices in static conditions, dynamic conditions, and also compared to these commercial insert systems. With our devices, we got in static, we got similar results as the commercial ones. In dynamic, we actually at 14 days, we started getting confluency, which may show that we may actually use lower seeding densities to get the same results. But this is work that needs to be further developed in the future. In this case, we also use a different kind of membrane, which was easier to use, a nanofiber-based mesh, which is also very common in the tissue engineering field, which is where we come from. So looking back at the initial project, we got an extensive validation for human intestinal model, but not as good for fish intestinal model. And why? Well, it's quite simple. So for the human, it's so the membranes were produced in Ghent, assembled into our devices in Ghent, and tested in the same place. For fish, we had to ship the membranes and the devices separately because the membranes were too delicate to be shipped mounted into the devices to our partners. And there it was more difficult to assemble the devices. Fair enough. So because of this, there was a considerable delay in getting the validation for fish. And also, this project wouldn't last forever. So there was at some point a decision to make on how to proceed. So the consortium at some point decided to switch to a plan B and just use standard commercial inserts to do the remaining of the work. And our colleagues will tell you all about it in the next talks. So for us as a company, there are many opportunities for the future. We're very happy with the work that we did. We have actually a new collaboration, among others, with CIMAR, actually with Louisa will be speaking in the afternoon. We actually have 3 new funded PhD projects, one together with Louisa for fish with microbiome integrated, and also for human as well. All kinds of tissue and organ interfaces like the gut, brain axis, colorectal cancer, its relationship with blood clotting, even study of Alzheimer's. So, and also to fund all of this, we applied for 2 EIC transition projects, one for fish, one for human. Hopefully, it will be a positive outcome. And next year, we actually plan to release 3 product families in the market. One of them based on the adjustable insert systems for any kind of application. Perfusion lids, also the same. We're very much happy to ship this to everyone, everywhere in the world for any kind of application. And then these electrospun membranes, which are very well known for being very good for cell growth because their architecture mimics the extracellular matrix of tissues. And we actually, in the meanwhile, developed a method to modify these membranes to actually also have this villi-like architecture in 3D, which is great. And we will be validating that in the coming years and then release that into the market as well as some more advanced membranes as well. Just to finish, some lessons learned from this adventure in fish AI. First of all, if you're trying to develop a device for researchers to use, don't try to force the user to adapt to your device. That doesn't work. It's a waste of time. Instead, just adapt the device right away to what the user wants and needs. Biology-focused scientists, unfortunately, in general, are not so much specialized in engineering, and they don't have to. It's not their job. So, and if you find someone like that, keep them. They're very valuable. And so that those are exceptions, okay? So the best thing to do is keep everything extremely simple, extremely obvious, as easy as possible. Start from existing concepts, so people that want to do this, use these devices know. So basically, they're standard operating procedures, and go from there. So from there, you can come up with slightly different ideas, step by step, upgrading the devices that you are developing and upscaling them as well. And that's all from my side. Happy to answer any questions that you may have. Thank you.
Thank you very much, Pedro. We have a few minutes for a few questions, and I would like to start with one coming here online that I will just read out loud from one of the online attendees, which is referring to what you described with these human cell studies and applications. And the question is, which of the information derived from these human cell studies that you did do you think can be applied to fish during these different approaches that was initially the plan of the project?
Well, I mean, the first main information is that this membrane and that this device, combined or separately, they work as models. That's the main conclusion of what we did. Of course, human and fish are not the same, and I'm not trying to say that. And in this project, it was unfortunately not possible to fully demonstrate and validate for fish, but that is something to do in the future. And I'm sure that sooner or later, it will be a usable model as well, and I would say quite valuable model in the fish industry as well.